Hello, my name is Ditte. I would like to present my poster titled Vascular Effects in Animals After Oral Exposure to Titanium Dioxide. And the in vivo studies I'm presenting here are part of my PhD project, which also deals with in vitro studies and a human intervention study. I have not conducted these studies yet, so unfortunately I don't have any results to show you. But the background is that in the recent years there are an increasing amount of particles produced and therefore an increasing human exposure. And nanoparticles are particles that are sized in one dimension in the nano range of 1 nanometers to 100 nanometers. And in this size the particles have some different chemical properties because of the increased surface to volume ratio. There are plenty of studies that are looking at pulmonary exposure to ambient air particles or engineered nanoparticles. They show an association with atherosclerosis. And the effect is thought to be through generation of oxidative stress and inflammation, which are believed to have a key role in development of endothelial dysfunction and also development of atherosclerosis. In contrast to these pulmonary studies, there are little knowledge about oral uptake of particles. The GI tract is also a very important route for exposure to particles, either through ingestions of the particles or via inhalation, inhaled particles that are moved from the lungs by the mucociliary clearing system to the GI tract. In my study, I will look at possible health effects of oral exposure to titanium dioxide. And titanium dioxide is used in a wide range of applications. I have some pictures here. You can see it's used in paint, in sunscreens, also in toothpaste and a lot of pharmaceutical products, and also in food products. I will be looking at food-grade titanium dioxide, which contains around 36% nanoparticles. And my experimental design, I have two studies. Study 1, where I will assess vasomotor function in the diet-induced obese rat. And this rat is fed a high-fat calorie diet for 10 weeks before entering the study. Then at study entry, I will, by oral gavage, expose the animals for titanium dioxide once a week for 10 weeks. And in study 2, which is pictured here, I will look at plaque progression in the ApoE knockout mice, which is a model for human atherosclerosis. And the mice is exposed to titanium dioxide once a week for 20 weeks. And my endpoints in study 1 is vasomotor function, which I assess by mounting vessel segments of the aorta in a myograph. And here I can add different vasoactive pharmaceuticals, for example, acetylcholine, which can, where we can look at endothelial induced vasorelaxation. And in study 2, I will look at plaque progression in the ApoE mouse. Immediately after cervical dislocation, I will cut out the aorta and look for plaques. Along with this, I will look at some biomarkers for oxidative stress, tetrahydrobiopterin, ascorbate, and malonaldehyde, as well as some pro-inflammatory markers. So with this project, I hope we can elucidate some mechanism behind particle-generated vascular dysfunction. Thank you.